So we're picking up talking about action potentials. Action potentials are your neuron's way of sending a signal. Remember, action potentials are traveling from the cell body the length of the axon to the axon terminal. Now, one of the things that's important to know about action potentials is that we call them an all or none response. What that means is that the size of an action potential doesn't change. So that graph that we looked at, if that was what it looked like for a neuron, that should be what it always looks like for a neuron with those same values. If the threshold is reached, then there's enough potential for action to happen. So if the cell body sends the axon a signal that depolarizes the membrane enough to cause the sodium channels to open, then we're going to start an action potential, and it's going to travel the length of the axon. If the stimulus is stronger, so let's say we're talking about light and the light was brighter, or sound and the sound was louder, or touch and the touch was harder, rather than having a bigger action potential so that your nervous system could interpret that the stimulus was stronger, it's the frequency of action potentials that would change. So frequency here is the key word. We're talking about how often they happen, but not the size. So we're going to take a look at just a little example here. Let's say that we have a stimulus. Maybe the stimulus is touch, or maybe the stimulus is sound. Whatever the stimulus is, it's increasing. We can see down here. Now, I'm going to write what the membrane potential is in millivolts. So when the stimulus is 1, the membrane is negative 70 millivolts. So it's still at rest, nothing happened. When we increase the stimulus to 2, membrane is still at negative 70. All of these units will be millivolts, so I don't have to write them again. Now we're going to say that stimulus at 3, that's the threshold. That's how much stimulus that neuron needs in order to start sending an action potential. As soon as we reach the threshold, where we're saying this is the threshold, we're going to get an action potential. And our action potential is going to depolarize the membrane up to positive 35. It's going to give us a full-size action potential. If I increase the stimulus to 4 or 5 millivolts, I'm not going to have a bigger action potential. The maximum point in my action potential will continue to be about positive 35 millivolts. So the amount of sodium that comes in and out of the cell in any one action potential isn't going to change. Now here's what will change. If we were to take a look at how many action potentials there were, we would say that there were 0 action potentials here, 0 action potentials here. Now, when we're talking about frequency, we're usually comparing a number to a time. So maybe we'll talk about action potentials per second. So once we hit the threshold, maybe we had 1 action potential every second. When we increased the stimulus to 4, now maybe we had 3 action potentials every second. When we increased the stimulus to 5, now maybe we had 7 action potentials per second. So we're not changing how big the action potential is, we're changing how many action potentials there are. So the way that your nervous system detects the strength of a stimulus isn't by how big each individual nerve impulse is, it's how many nerve impulses there are over a period of time. Now, as we follow an action potential from the beginning of the axon all the way to the end, the impulse does not stop once it's started, not unless there's something wrong with the neuron. It will travel the entire length of the axon. So it's all or none. Either it happens and it travels from the cell body through the axon, or it doesn't. The impulse always travels in one direction, so always from the dendrite to the soma to the axon terminals at the end of it. Between two neurons, so if I'm talking about the one neuron communicating with another, it comes from the axon to the dendrite of the next neuron. Now, as an action potential is propagating over the length of an axon, sodium ions that entered during the action potential diffuse in the axon. So I'm not saying more of them enter. I'm saying that now that they're inside of the axon, they start moving down the length of the axon. Ions always want to move from a place where they are more concentrated to where they are less concentrated. So at the point where they came into the axon, there's a whole bunch of them, and they're particles. They don't have brains. They don't know where they're supposed to go. They start moving down the length of the axon. When they get to the next node of Ranvier, that bit of positive charge can cause depolarization. That can be enough to open the sodium channels and let another action potential happen. Now, I'm always thinking about things going from left to right because that's how we would read. And on the picture I'm going to show you in just a second, that's what's happening. But if we're imagining an action potential going from the left to the right on a picture, where the action potential just happened, the membrane is refractory. That means even though it might be depolarized a little bit, it's not enough to start another action potential, and it prevents the impulse from going backwards. The conduction of a nerve impulse is called saltatory conduction. It's called saltatory conduction because the impulse jumps from one node to another. Now this is true for myelinated neurons. If we had an unmyelinated neuron, then we wouldn't have saltatory conduction. There's a disorder that's directly related to this process. It's multiple sclerosis, so MS. In MS, we see demyelination, so the destruction of myelin on central nervous system neurons. So nerve impulses decrease in frequency or in speed because myelinated neurons transmit the signal more quickly. Now here's the picture I was talking about. We're looking at a neuron. There's the dendrites in the cell body. Here's the axon. Each of these little sort of oval-shaped things is a piece of myelin sheath, and the spaces in between them are nodes of Ranvier. If we take this little section and we zoom in and blow it up, we see that there is a node of Ranvier right on the edge. Hopefully you can see sodium channels are open and sodium is rushing in. That's causing an action potential. Now those sodium ions, like I said, start diffusing down the axon. Some of them, sure, will get pumped back out by the pump, but some of them will make their way down the axon. As they do, when they get to the next node of Ranvier, it can bring it to threshold. It can cause enough depolarization that the sodium channels here are stimulated to open and let sodium rush in, causing another action potential, and it would continue along the length of the axon. Now, when we get to the end of the axon terminal, we see a synapse. So when we're looking at this picture here, the synapse is the space, so you can see it right here, between the end of one neuron and the dendrite of the next one. So this part here at the beginning, here, that would be the axon terminal. And you can see over here, this is the dendrite. So this whole pink section down here would be one piece of a branch of dendrite on the following neuron. Now, when we're talking about these two neurons, we need to know which one is sending the signal and which one is receiving the signal. The one that's sending the signal over here, this is the presynaptic neuron, and you can see the word presynaptic down here. The one that is receiving the signal, so the one whose dendrites are in the picture, that is called the postsynaptic neuron. And that's how we refer to the two neurons that make up the synapse. Now, this is a picture of a synapse of two neurons. A synapse could be between two neurons or between a neuron and an effector. I'm showing you an example between two neurons. The word synapse means link, or junction point. We could have a neuromuscular junction. Those are pretty typical in the body. This is anywhere there's a synapse between a motor neuron and a muscle cell. The space between the two neurons is specifically called the synaptic cleft. So that little bit of space that's in between them. So they're not physically touching each other. There's a little gap of space. So what happens? The influx of ions, so the nerve impulse, arrives at the presynaptic terminal. So it would be coming from this direction, from the axon that would be up here. And it causes the opening of some calcium channels. Now, when calcium channels are opened, they stimulate the cell to start this process of releasing chemical messengers. So chemical messengers in this context are called neurotransmitters. They are the things that are going to take the signal from one neuron to the next. So you can see all of these little dots here are neurotransmitters. You can see some of them in the synaptic cleft, and you can also see some of them over here in the presynaptic neuron inside of little vesicles. Now there are some synaptic vesicles, and you can see them right here. They are the things in the presynaptic neuron that contain neurotransmitters. If you're unsure of which neuron is the presynaptic neuron, look for the one with the vesicles, and that'll be it. These vesicles will fuse, like you see over here, with the axon membrane. So this is a process called exocytosis, which you would have learned about in previous science classes. But essentially the vesicle, which is made up of a membrane, will fuse with the cell membrane, causing the contents to be released into the synaptic cleft. Now, like I said, that's where the calcium played its, or had its role. There's some other structures here. I'll just mention them. Those are mitochondria, need energy for this process to happen. It does not take a ton of time, between 0.5 and 1 millisecond, for the neurotransmitter to reach the dendrite of the postsynaptic neuron or the effector membrane. So remember where the dendrites are, that's the postsynaptic neuron. Here's a little receptor. That's something that the neurotransmitter could attach to or bind to. So neurotransmitters bind to receptor proteins. They can be different shapes and sizes, but there needs to be a structure or a protein where the neurotransmitter can attach. If they are exciters, so if they are excitatory, they cause sodium channels to open and they depolarize the postsynaptic membrane. So they do something similar to what happens at the beginning of an action potential. If the neurotransmitter is inhibitory, it causes potassium channels to open. So if you think about an action potential, potassium wants to leave the cell. That makes the dendrite even more negative than it was before. So excitatory neurotransmitters let sodium in, inhibitory neurotransmitters let potassium out. If the threshold is reached, so remember the threshold is talking about depolarization. So if enough positive ions like sodium go in, then that will trigger the opening of the sodium channels, and then an action potential will be initiated. Now because, remember there are lots of dendrites that could all be receiving different signals, the cell body integrates all of the signals, and the combined effect will decide if for the neuron as a whole, we've met the threshold, and now it's going to send an action potential down its axon or not. Now there are lots of neurotransmitters. We're going to focus on just a few. The most important one that you need to know about for right now is acetylcholine. Acetylcholine, or ACH for short, is a neurotransmitter that crosses a neuromuscular junction. So this is the neurotransmitter that communicates between a neuron and a muscle. So if you look over here, you can see the presynaptic neuron. You can see where the receptors are. Now, in this case, it's not the postsynaptic neuron, but rather the muscle or the effector. You can see that there are receptor proteins for the neurotransmitter to bind to. You can see that there are vesicles in the presynaptic neuron and that they're fusing with the membrane through exocytosis. Now, for most neurotransmitters, we need a way to get rid of them. We don't want them to stay in the synapse. And in this case, the case of acetylcholine, keep your muscles in a state of permanent excitement or permanent contraction. So there's a mechanism to help get rid of them. There's an enzyme called cholinesterase or acetylcholinesterase. So it's this little red triangle here, little enzyme. It will help acetylcholine, the neurotransmitter, break down. Often when it breaks down, it breaks into two or more component pieces. So if you see here, there's these little triangles and these little circles. Those are the acetyl-CoA and choline that together make up acetylcholine. The enzyme causes them to break apart so that they stop attaching to the receptor and they stop sending the signal. Now, in a lot of cases, these component molecules here can go through a reuptake process. So the presynaptic neuron can retake them or reabsorb them so that they can be put back together and used again. Now, a lot of drugs, and that's what your assignment will be about, is drugs, will affect the way a neurotransmitter communicates between one neuron and the next. For example, there's a neurotoxic gas called sarin. It destroys this function, so it doesn't allow cholinesterase to do its job. That causes acetylcholine, the neurotransmitter, to stay in the synapse, continuously binding to receptors, causing a constant state of contraction. This is toxic because if neurons continually cause muscles to contract, bad things are going to happen. This particular neurotoxic substance is the basis for a lot of insecticides. There are other neurotransmitters. You can see some details on page 382. Dopamine is one you hear about a lot, especially when you're talking about illegal recreational drugs, because it is the neurotransmitter not only that controls movement, but also that causes feelings of pleasure. Serotonin is another neurotransmitter. It has a lot to do with sensory and temperature perception. Endorphins are something you might have heard of. They are a type of neurotransmitter that cause pain relief. So your body produces these naturally. Norepinephrine or epinephrine are some substances that we'll talk about a little bit later on in the course when we talk about stress and how your body responds to stress. But these are just some other examples. Just like acetylcholine, enzymes will help remove them from the synapse, but they are chemicals that communicate between one neuron and the next, or between one neuron and another effector cell like a muscle.